This is the second video discussing skeletal muscle tissue. In it, we're going to finish talking about the structure of skeletal muscle, and then we'll begin to talk about excitation-contraction coupling. So now we need to look at how we make up the thick and the thin filaments, and we're going to start with the thick filament. The thick filament is composed of approximately 300 myosin heavy chains, and that's what you can see here. These are the myosin heavy chains. And you can see that they have a very distinctive structure. They've got the typical alpha helix that we expect here for proteins, and then you can see it's kind of double-headed at this end. And so people like to compare this to a double-headed hockey stick or a double-headed golf club or a variety of different things. But there are some important parts here that we need to look at. And then again, we'll look at how we put together all of these myosin heavy chains to create the thick filament. And so that's what we're seeing here is indeed the thick filament. Here's a slightly different depiction of this end of the myosin up here. And there are some very important characteristics that you need to be familiar with and you need to understand. Number one, you can see that there's a binding site here for ATP, and there's also an ATPase, which will very quickly, almost immediately, hydrolyze the ATP to ADP and inorganic phosphate. The other nice thing is that you can actually see a photomicrograph of a couple of molecules of myosin, and here you can see the helix, and here you can see the two heads. Here's a different depiction of that. The other thing that's important here is that not only do we have the binding sites for the ATP, but we also have binding sites for actin. And so we have an actin binding site on each one of these heads. So very important. 2 ATP binding sites, 2 actin binding sites that we have here as we look at this. And we start to build these into the thick filament. I don't like this depiction of a sarcomere, but I do kind of like this depiction of the thick filament. And you look carefully here and you can kind of make out some of the individual myosin molecules that are going into building this thick filament. And then what we have here down the middle is we have the M band, and this is made up of a variety of proteins, including myomycin, M protein, and obscurin. And at this point, it's unclear whether or not there are differences in the M-line protein between different fiber types as we look at this. Here's a slightly different depiction of the individual myosin molecule. But again, you can see both the actin binding sites and the ATP binding sites. The other important part of the individual myosin is that right here behind the head, we have a hinge, and back a little bit further along the tail, we have a hinge. And the hinge here allows this part of the myosin to move up towards the thin filament. That's what it allows it to do, move up and actually grab a hold of the thin filament. And then the hinge here actually allows these heads to kind of bend and pull so that all the heads are pulling that thin filament that way. All the heads on this side are pulling the thin filament that way. And as you can imagine, what that's trying to do is it's trying to bring these Z discs of the sarcomere closer together as this thing contracts. Again, what we have here in the middle is the M band and made up of a variety of proteins, myomycin, M protein, obscurin, perhaps even some others that we'll learn about a little bit later on. Again, just to reiterate, we have the actin binding sites and the ATP binding sites on the heads of the myosin molecules. And again, I'm going to reiterate that the ATP essentially is hydrolyzed to ADP and phosphate as soon as it binds to the myosin. Okay, and then we have the two hinges that are very important in the function of the muscle. Here you can see the thick filaments. Maybe you remember what this is. I hope you do. But there you can see the thick filaments with the M band down the middle, and this is the titin. And the titin is, again, anchored here at the M band as we look at this. So here's a slightly different depiction of the thick filament. That's what we have. We have the thick filament. And those are the individuals, the individual myosins that make up the thick filament. And again, they're reminding you of the binding sites that we have for actin and ATP. And also the ATPase that exists there. I'll also point out to you, notice that as we work our way around, the cross bridges are coming out at various angles. How many thin filaments surround this thick filament? And I hope that you said 6 of them. And so that's what's going on here. So this thick filament is made up of approximately 300 of these guys. And again, they rotate as they move down around. And the M band here and here is made up of these proteins, myomesin, M protein, obscurin. All right, now we'll move on and we'll look at the structure of the thin filament. Here is the thin filament all put together, but you can see that the thin filament is made up of the protein actin. Here is an individual actin molecule, and you look at it and it looks a little bit like maybe a moldy olive. And so here's the olive part, and this is where the pimento of the olive would go. Some people say it looks like a black-eyed pea. I don't care. The important point is that the actin has this active site. This is where the myosin cross bridge, remember it has a binding site for actin, is actually going to grab a hold of this. And then we have a new compound which we call actomyosin. We take these individual molecules of actin and we start to build them into F-actin, and then we take 2 F-actins and we create a helix, and now finally we have the actin in the form that it will be used when we build the thin filament here. Actin and myosin are referred to as structural proteins. The other part we have here is we have troponin. You can see the troponin here. There are 3 parts to the troponin. Troponin C, which as we'll find later on binds to calcium. Troponin T, which binds to this tropomyosin, and troponin I, which some people consider the inhibitory subunit of the troponin. 3 troponin subunits. And then here you can see the tropomyosin lying along the length of the actin filament, and notice what's covered up here. And if you're paying attention, you notice that this covers up those active sites of the thin filament. Now, the troponin and the tropomyosin are known as regulatory proteins because they're regulating whether or not contraction can occur. And so here's just a different depiction for you. Here is the thin filament anchored in the Z disc. Okay, and here it is projecting outward. And so again, you can see the actin filament, and you can see the 3 troponin subunits, and you can see the tropomyosin that lies along the top of the active sites of this thin filament. And so the active sites are not available for the myosin to grab a hold of. Here's a slightly different depiction. And so again, the ATP is bound to the cross bridge and it's been hydrolyzed. Now there will be ADP and phosphate here as well. They've been a little bit lazy. All of these cross bridges, when the muscle are at rest, will have ADP on them because ATP has bound and been hydrolyzed, and so all of the cross bridges have ADP, not just the 2 that they've shown you. They've just shown you the 2 that line up facing the thin filament here. Remember that there are 6 thin filaments surrounding this thick filament. So thin filament with the F-actin, the tropomyosin, and the troponin. Now here we see that the active sites have been uncovered. The troponin and the tropomyosin have been moved out of the way. And you should remember from anatomy and physiology, that is what's called calcium disinhibition. Calcium comes in and it binds to the TNC. The troponin changes shape, and since it's attached to the tropomyosin, it pulls the tropomyosin out of the way. And so now the active site has been uncovered. Remember, we've got the 2 hinges. And the first hinge can actually bend and allow the myosin to reach up and grab a hold of the actin. We now have a new compound, actomyosin. actomyosin. And now we can bend at the second hinge right here, and it's going to pull in this direction. And all the myosin on this side of the sarcomere is going to pull in this direction. That means that the Z disc is over this way, the M band is over this way. And everything is going to try to pull the thin filament towards the M band in the middle of the sarcomere. Here again, we can see a nice depiction of the sarcomere. Here's the Z disc. There's a lot of protein in here. They're just showing you one of the early ones and one of the more important ones, alpha-actinin. And both the thin filament and the titin are anchored here at the Z disc. And here again, you can see that the titin is moving from the Z disc. Two of them come in and they kind of converge here at the thick filament, and then they move through and they're anchored here at the M band. And so there you can see that titin. This is another great photomicrograph of a sarcomere. At this point, I've never seen a photomicrograph of a sarcomere in which you can actually see the titin that is in there, but that's what you can see. Again, just a slightly different depiction showing you the titin and how it comes from the Z disc, kind of converges here at the thick filament. This one doesn't do as good a job. It almost makes it look as if the titin moves through the M band and continues on. And perhaps that's the case, but that's not what we believe right now. What we believe right now is that we have two titin to the right side, two different titin to the left side, and they're anchored here in the M band. That's what we believe at this point. The other thing that you should notice here is, let me see, we've introduced a new protein here associated with the thin filament, nebulin. And so you can see that nebulin 2 is anchored here at the Z disc, and it comes all the way out here to the very end of the actin. And what nebulin does, it's unclear. The function is nebulous, and that's where the term nebulin comes from. But we have good evidence to make us believe that it helps to set the length of the thin filament. It seems to act as a molecular ruler and set the length of the thin filament. When we use knockout mice and we get rid of the nebulin, then some of the thin filaments are too short, some are too long, and so the length is not quite where it's supposed to be. Again, here we can see the titin. Why is it all wound up like this? What does this represent? What does this represent? And so again, I hope you said that this is the spring-like region that we find in the I band for the titin. Okay, and then again, you can see the titin there. Titin, what does it do? Well, the ends again anchor at the M line and the Z disc, and we may learn as we study this better and get more detail that we're going to modify this a little bit. But at this point, that's what we believe. At one end, it actually interacts with the alpha-actinin here at the Z disc. It acts as a molecular spring in the I band region. And so it's what's primarily responsible for the elasticity of skeletal muscle. It also seems to keep the thick filament in register with the thin filaments. So it helps to perhaps serve a structural function as well as provide that elasticity, which is important. And here at the M band, it may interact with the myosin and the myomycin. Again, that's what data seems to suggest, but we'll have to wait and see. Here again, you can see the nebulin. The nebulin seems to be this thin filament ruler. And so it helps to determine that length of that thin filament. But there is some data that says it also plays a role in muscular contraction. At this point, we don't know exactly what that role is. And again, I will reiterate that initially it was referred to as nebulin because the function of that protein was nebulous. We didn't know what the heck it was. I'm going to mention one other set of proteins that we have here. These proteins actually seem to link the Z disc to the sarcolemma and to the extracellular matrix. These are called costameres. And the one that I'm going to mention to you is dystrophin, because after decades of research, we've finally found that there's a problem with the dystrophin in people who have muscular dystrophy. It's important that the tension developed by the sarcomeres be transmitted to the connective tissue sheaths. And it seems like these proteins, these costameres, are responsible for doing that. And so we may see the breakdown of those as people age as well. Now we're going to go ahead and we're going to talk about the membranes of the skeletal muscle. And these are critical, very, very important. 3 plasma membranes here. And they play a role in the process of excitation, contraction, coupling. That's what they do. Well, what are the membranes? Well, number one, sarcolemma. It is the membrane of the muscle cell itself. All of these are phospholipid bilayers. Number 2 is the transverse tubule. It actually is continuous with the sarcolemma, and it moves down at right angles to the sarcolemma, sometimes called the transverse axial tubular system. I may never say that again. And then we have the sarcoplasmic reticulum. And again, as I've told you a couple of times, this is specialized endoplasmic reticulum. Now, your book and other books talk about the terminal cisternae. People get confused. They think that it's a 4th membrane, but it's really just a part of the SR. And I don't care too much about the terminal cisternae. It doesn't help you in understanding what's going on here. You have to know the membranes of the skeletal muscle that are involved in excitation, contraction, coupling, and there they are right there, 1, 2, 3. And you also need to understand the protein channels that are in here and the pumps. These are important in altering membrane permeability to sodium, potassium, calcium, things like that. And so they play a critical role in excitation, contraction, coupling. And then the pumps, primarily sodium, potassium pumps, but also calcium pumps. And so they too are very, very, very important. Well, where are these skeletal muscle membranes? And before we look at that, I just want to remind you, do not confuse the membranes and the connective tissue sheaths. Every semester, somebody does that on the test. I ask for the connective tissue sheaths, I get the membranes, or vice versa. So don't confuse them. Here again are the membranes. And so what they've done here is they've reflected the sarcolemma here. That means that this is a muscle fiber. And they must have peeled off and taken away the endomysium. Don't forget that's a connective tissue, not a phospholipid bilayer. This is a phospholipid bilayer. Here you can see these purple pieces in between. Those are the transverse tubules. That's what they are. And here you can see the opening of transverse tubules. These are little holes in the sarcolemma, and they're the opening up of the transverse tubule. And then finally, this net-like stuff, that is the sarcoplasmic reticulum. And so it's the bluish stuff that you see here. And so sarcolemma, transverse tubule, sarcoplasmic reticulum are all phospholipid bilayers. That's what they are. Okay? And again, here you can see that terminal cisternae is just a part of the SR. Just a part of the SR. Here's a slightly different depiction. Hard to see the sarcoplasm here because they haven't reflected it. But at any rate, here you can see the T-tubules. And so again, little holes in the sarcolemma. That's what they are. It's like the hole on a golf green. And it goes down, and here we've begun to get into the transverse tubule. And so you can see it moves away at a right angle. Notice that the transverse tubule is completely surrounding the myofibrils. That's what this is. It's a myofibril. Here's a myofibril. And so the myofibrils are completely surrounded by the transverse tubule. Again, the sarcoplasmic reticulum here is in blue, and it's this net-like structure. Look right here. And so you can see that the myofibrils are also surrounded by sarcoplasmic reticulum. So the myofibrils are completely surrounded by transverse tubules, which are continuous with the sarcolemma, and sarcoplasmic reticulum. Okay, this part of the sarcoplasmic reticulum is the terminal cisternae. That's where the majority of the calcium is located. But it's just a part of the sarcoplasmic reticulum. Here's a third depiction. Again, it's hard for us to see the sarcolemma. It's this beige-like thing that you see here. Now they're showing you that this guy is multinucleated. This is part of a muscle fiber, and we've cut away the sarcolemma. Now in this case, again, the transverse tubule is yellow, but now the sarcoplasmic reticulum is greenish. And so a variety of pictures. I show you this picture just so that, again, you can see that the transverse tubule is connected to the sarcolemma and moves away from it at roughly right angles. And then it's going to surround every single one of the myofibrils. And so there again, you see those three connective tissues, which are important in the structure and function of the muscle. The sarcoplasmic reticulum, again, is specialized endoplasmic reticulum. Here it is. And so the sarcoplasmic reticulum contains lots and lots of calcium, which is the inside of the SR. Well, it's over here. And the way that I know that for sure is because here's the calcium pump. The pumps are pumping calcium back into the sarcoplasmic reticulum after muscle contraction has occurred. These guys that you see here, those are voltage-gated calcium channels. Voltage-gated. Well, what's stored in and released from the SR? Calcium. There's calcium. And that can escape by going down its concentration gradient from inside the SR to the cytoplasm of the muscle fiber. Membrane proteins, well, I just told you, voltage-gated calcium channels and calcium pumps. And so again, what do these guys need to use that isn't shown here? Oh, they need energy. They need to hydrolyze ATP to get the energy to pump the calcium back into the inside of the sarcoplasmic reticulum. What's true of the sarcolemma and the transverse tubule? Well, they're continuous one with one another. That's what's true of those two. What is the primary function of skeletal muscle? Generate tension. It contracts, it generates tension. It doesn't always shorten, but that's what it's trying to do. Well, what is the first thing we need to do to get a muscle to contract? Well, we need to activate it. That's what we need to do. Contraction of a muscle is a mechanical event. Activation of a muscle is an electrical event, and the electrical event has to precede the mechanical event, and so that's what leads to excitation-contraction coupling, and you need to understand what this is and what's going on here. And so here we can see the beginning of that. Here we can see the alpha motor neuron. Okay, and here is the synaptic knob with all of those synaptic vesicles in here. Here's the cleft. And here is the specialized sarcolemma, which is motor end plate. And so there you can see the receptors for the neurotransmitter, which for a muscle is always, always, always acetylcholine. And so there you can see that. So what are the three parts of the neuromuscular junction? Well, number one, the synaptic knob. Number two, the synaptic cleft. And number three, the motor end plate. And so here these parts are important. This is the action potential that's arriving here, travels down the telodendria, gets to the synaptic knob, and that's what opens up these voltage-gated calcium channels. Calcium is in higher concentration outside the cell, and so it moves into the inside. And you should remember from our discussion of chemical synapses that that's what causes migration and docking of these synaptic vesicles. The vesicles open up and release acetylcholine via exocytosis. And you know exocytosis is an active process, so there also are going to be a lot of mitochondria in here. The neurotransmitter diffuses across and it binds with these nicotinic cholinergic receptors to open up the ligand-gated ion channels. And so the ligand-gated ion channels open up. Again, the motor end plate is specialized sarcolemma, and that's where the ligand-gated ion channels are found. This is just a different depiction of the neuromuscular junction. And so here is our motor end plate, which again is a specialized chemical synapse. These are the myofibrils underneath. Here is the specialized sarcolemma. This is phospholipid bilayer with ligand-gated ion channels throughout. The green stuff is what? Acetylcholine. And so the green stuff is acetylcholine. And so again, we'll go through the process, but you should understand it. The action potential arrives here. Voltage-gated calcium channels open. Calcium enters. The synaptic vesicles will migrate and dock. The acetylcholine is released via exocytosis, and it moves across and it binds with the nicotinic receptors. As a result of that, the ligand-gated ion channels open up, and you can see that potassium leaves, but more sodium is coming in. And as a result, what we get is we get an end plate potential. And it's that end plate potential that's going to move along, and it's going to stimulate the opening of voltage-gated sodium channels and voltage-gated potassium channels that are just outside of this neuromuscular junction. And so again, you have to remember that these proteins are both the ACH receptor and the ligand-gated ion channel in one. And so then to the side, we have the voltage-gated sodium channels. You can see that it takes 2 acetylcholine to open up the ligand-gated ion channel. And again, as the channel opens, we've altered the membrane permeability. Sodium comes in. We create an end plate potential, which is just simply a specialized EPSP. And it's that EPSP which moves along and stimulates the opening of the voltage-gated sodium and voltage-gated potassium channels, which will lead to an action potential. Here we can see a real live neuromuscular junction. And then again, this is what we've talked about before, and so you should maybe pause it and review this on your own. And that brings us to the end of video number 2, and in video number 3 we'll finish up with our discussion of excitation-contraction coupling.